Have you ever wondered about the secrets hidden within our DNA? It's a question that has intrigued scientists for decades, and continues to do so today. DNA, or deoxyribonucleic acid, is often likened to a blueprint or a recipe book, containing all the instructions necessary for life. At the heart of DNA's structure are its molecules, composed of two strands that twist together to form a double helix. It's a design that's as beautiful as it is complex, and it's made up of basic components called nucleotides. Each nucleotide is a tiny powerhouse consisting of a sugar molecule, a phosphate group, and a nitrogenous base. There are 4 types of these bases: adenine, thymine, guanine, and cytosine, often abbreviated to A, T, G, and C. These nucleotides line up in a specific order to form a chain, and it's this sequence that provides the instructions for building and maintaining an organism. But how do these chains come together? Well, it's a bit like a puzzle. The adenine base on one strand always pairs with thymine on the other, and guanine always pairs with cytosine. This is known as base pairing, and it's this process that enables the DNA strands to hold together and form the familiar double helix structure. But the DNA chain isn't just a static structure, it's dynamic, constantly unzipping and re-zipping in a dance of life. It's this dance that allows DNA to replicate itself, to repair damage, and to pass on genetic information to the next generation. Understanding the structure of DNA and the role of nucleotides has been a crucial step in unraveling the mysteries of genetics. It's a journey of discovery that has uncovered the fundamental building blocks of life, and has paved the way for advances in fields as diverse as medicine, forensics, and agriculture. This intricate structure is the blueprint of life, encoding our genetic information. It's a testament to the wonder of nature, and a reminder of just how much we still have to learn about the secrets hidden within our DNA. Genes, the genetic information carriers, hold the key to our traits and development. Now, what exactly are genes? Picture a long ladder twisted into a spiral. This is your DNA, the blueprint of life. Genes are segments of this DNA, each carrying instructions for building a specific protein in your body. These proteins then go on to perform a multitude of functions, ultimately shaping your physical traits and biological functions. Genetic information is carried from one generation to the next through the process of reproduction. The genes from both parents combine to create a unique set of genes for the offspring. This genetic combination determines everything from the color of your eyes to your susceptibility to certain diseases. The impact of genes on phenotype, which is the set of observable characteristics and organism development, is profound. They influence how organisms grow, develop, and adapt to their environment. These tiny segments of DNA dictate the traits we inherit, making us unique. The foundation of genetics was laid by Gregor Mendel. This monk, turned scientist, performed basic yet revolutionary work in the mid-19th century. His playground? A humble pea plant garden. Through meticulous crossbreeding experiments, he observed patterns that led to the formulation of the fundamental principles of inheritance, now known as Mendel's laws. The first law, the law of segregation, states that an individual inherits one allele from each parent for each trait. The second, the law of independent assortment, proposes that the inheritance of one trait does not affect the inheritance of another. These laws provide the framework for understanding how our traits are passed down from our ancestors. Despite their simplicity, Mendel's laws are the cornerstone of genetics, a key to understanding the complex puzzle of life. Thanks to Mendel's pea plants and his keen observations, we've begun to unravel the mystery of nature's code. Mendel's laws unravel the mystery of how traits are passed on from generation to generation. DNA replication, a crucial process during cell division, ensures the continuity of life. This fundamental biological procedure allows cells to divide and grow, while maintaining the genetic integrity of the original cell. Think of it as a high-stakes game of copy and paste. The DNA molecule unzips, revealing its 2 strands. Each strand serves as a template, a blueprint if you will, for constructing a new complementary strand. Enzymes, the little workers of the cell, scurry along each strand, reading the genetic code. They match each DNA base, adenine with thymine and cytosine with guanine, to create a mirror image. As the enzymes zip along, they join these bases together, forming a new strand that pairs perfectly with the original. This process creates 2 identical DNA molecules, each a clone of the original, ready to be packaged into new cells. Through replication, DNA ensures life's continuity, passing on genetic information to the next generation. Genetic mutations, though often feared, are essential drivers of evolution. These changes to the genetic code, the DNA, happen when an error is made as the DNA is copied during cell division. This could be like a typographical error in a manuscript, changing one letter, deleting a letter, or even adding extra letters. These mutations might seem like mistakes, but they're a vital part of nature's grand design. You see, these tiny changes can lead to significant variations in an organism's traits. For instance, a mutation might cause a bird to develop a beak shape more suited to its environment, allowing it to access food resources more efficiently than its peers. But how does a mere mutation translate into a species-wide trait? The answer lies in the concept of natural selection, a process that Charles Darwin famously described as survival of the fittest. In our bird example, the mutation gives the bird an advantage, and it's more likely to survive and reproduce, passing on this beneficial mutation to its offspring. Over generations, this trait becomes more common in the population, leading to evolution. Genetic variation, the differences in DNA sequence among individuals, plays a critical role in this process. It's the raw material upon which natural selection acts. More genetic variation means more opportunities for beneficial mutations to occur, making the species adaptable to various environmental changes. So, while genetic mutations might seem like nature's typos, they are anything but. They're the engine driving the diversity of life on Earth. From the color of a butterfly's wings to the shape of a cheetah's spots, mutations have sculpted the natural world in countless beautiful ways. And it's not just about physical traits. Mutations can lead to behavioral changes too, shaping how animals interact with their environment and each other, influencing everything from feeding habits to mating rituals. Mutations, nature's tool for diversity, are the driving force behind the never-ending process of evolution. The future of genetics holds exciting possibilities, transforming our understanding of life. Our journey into the realm of genetics has been nothing short of a roller coaster ride, replete with breakthroughs, setbacks, and surprises. The advent of genome sequencing and genetic engineering technologies have revolutionized our understanding of the genetic blueprint that makes us who we are. But what does the future hold in this exciting field of science? The recent years have witnessed a surge in genetic research, with scientists making headway in areas previously thought to be out of reach. For instance, the ability to edit genes using CRISPR-Cas9 technology has opened up a world of possibilities, from curing genetic diseases to potentially creating designer organisms. The future could see us perfecting these techniques, leading to more precise and targeted genetic modifications. In addition, the field of epigenetics, which studies how environmental factors can influence gene expression, is set to take center stage in the years to come. We are beginning to understand that our genes are not fixed entities but rather dynamic elements that can be influenced by our surroundings. This could potentially lead to breakthroughs in how we approach the treatment of diseases, by not just focusing on the genetic code but also how it is expressed. Moreover, the advancement in technology has made genome sequencing more accessible and affordable. This could lead to a future where personalized medicine becomes the norm. By understanding an individual's unique genetic makeup, doctors could prescribe treatments that are specifically tailored to their genetic profile, improving their effectiveness and reducing potential side effects. The potential of genetics extends beyond medicine. As we continue to map the human genome, we can gain insights into our ancestry and evolution. In the future, we might be able to trace our lineage back to the earliest humans, or even pinpoint the genetic markers that set us apart from our closest primate relatives. But as we charge forth into this brave new world of genetics, we must also consider the ethical implications of our actions. How do we balance the potential benefits of genetic modification with the risks? How do we ensure the privacy and security of individuals' genetic data? These are questions that we must grapple with as we continue to push the boundaries of what's possible. The future of genetics is a vast and uncharted territory, full of promise and potential pitfalls. But one thing is certain, the discoveries and advancements made in this field will continue to transform our understanding of life in ways we can't even begin to imagine. As we continue to unravel the mysteries of DNA, we stand on the brink of a new era in genetic science. The possibilities are endless, and the future is bright. So, here's to the future of genetics. A future that promises to redefine life as we know it.